Hi, welcome to Enzo Network. Today you will learn everything about biotechnology, its inventions, advantages, and risks. It all started in 1600s. A Dutch scientist named Antonie van Leeuwenhoek looked through a microscope and discovered something humanity had never seen before: microorganisms. That discovery changed biology forever. But scientists still didn't know what are the uses of microorganisms. And after 200 years, that changed. A French scientist, Louis Pasteur, proved that microorganisms were responsible for fermentation, and it laid the foundation for modern medicine. Because of Louis Pasteur, for the first time, humanity began to understand that invisible living organisms could shape our world. This is the birth of genetics. In the 1860s, an Austrian scientist named Gregor Mendel conducted experiments using thousands of pea plants. His findings revealed that traits are passed down through biological instructions we now call genes. The effect was enormous. They began to realize that biological information follows rules. But there was still one major question: how are these instructions created and stored? That answer arrived in 1953. James Watson, Francis Crick, Rosalind Franklin, and Maurice Wilkins helped uncover the structure of DNA. For the first time, scientists understood the molecule carrying the instructions of life. This is the foundation of biotech. DNA became biology's equivalent of computer code, and once you understand the code, a new possibility emerges. But can we change DNA? In the 1970s, scientists developed genetic engineering. Instead of simply studying DNA, they began modifying it. Researchers learned how to cut DNA, copy DNA, and transfer genes between organisms. This was a turning point. Biology was no longer just a science of observation, it was becoming a science of engineering. Scientists inserted the human insulin gene, then produced insulin at scale. Today, millions of diabetes patients rely on medicines made possible by this breakthrough. The results demonstrated a powerful idea. Living organisms could become biological factories. But scientists wanted to go further. By the 1990s, researchers knew DNA contained life's instructions. Yet they still didn't know the complete sequence. This led to one of the largest scientific collaborations ever attempted: the Human Genome Project. Beginning in 1990, thousands of scientists worked together to map the entire human genome. The project revealed that humans possess roughly 20,000 genes and are approximately 99.9% genetically identical. The impact was immediate. Researchers gained a complete map of the human instruction manual. That accelerated drug development, personalized medicines. But the biggest breakthrough was still ahead. In 2012, scientists adapted a bacterial defense system known as CRISPR into a gene editing tool. For the first time, DNA could be edited with unprecedented precision. Think of DNA as a document. CRISPR acts like a search and replace function. Scientists can locate a genetic sequence, remove it, modify it, or replace it. The success of the project spread all around the world. Now, genetic diseases caused by a single mutation may eventually be corrected at their source. Researchers are already exploring treatments for sickle cell disease, inherited disorders, and certain cancers. But important part is, biotechnology isn't only transforming in medicine. Agriculture is another major frontier. Because global population moves toward 10 billion people, food demand continues to rise. At the same time, climate change is increasing droughts, floods, and crop failures, and biotechnology have solutions. Scientists are developing crops that require less water, resist pests, and produce higher yields. The goal is not simply to grow more food. It's to grow food more efficiently and sustainably. But now you need to understand, yet every powerful technology comes with risks. Gene editing raises difficult ethical questions. Should humans modify embryos? Who decides which genetic changes are acceptable? What happens if powerful technologies are misused? Example: if gene editing technologies like CRISPR become capable of changing embryos, they could potentially be used not only to prevent serious diseases, but also to select certain physical or biological traits. This raises questions such as, this could increase social inequality, dangerous biological experiments on humans, unauthorized access to sensitive health information. The challenge is balancing innovation with responsibility, because biotechnology is different from previous technologies. It doesn't simply change machines, it changes living systems. But one thing is sure, the decisions we make over the next few decades may determine the future of our own species. Thanks for watching.